Good morning students, welcome to my channel. Today we are going to discuss about meiosis. In my previous handwritten video I have already discussed about that mitotic cell division occurs in somatic cells, but this is one of the complicated cell division process that occurs in reproductive cells, that is called as meiosis. The term meiosis was coined by a farmer. It occurs in reproductive cells. It is otherwise called as a reduction division, as the chromosome number of parent cell become haploid into two daughter cell. When the parent cell containing that diploid number of chromosome, that is 2n, it produces 4 daughter cells having haploid number of chromosomes, that is n. So meiosis is a complicated division which is further divided into two types. First one is meiosis 1 and second one is meiosis 2. The meiosis 1 is otherwise called as heterotypic division, whereas meiosis 2 is otherwise called as homotypic division. The heterotypic division or meiosis 1, it is the reduction division by which the parent cell reduces its number of chromosome into haploid. But the homotypic or equational division just equivalent to that of mitosis. So the chromosome number remain unchanged as it is in parent cells, and two daughter cells are formed from each parent cell. So the cell that undergo meiosis, that is called as meiocytes. That cell is called as meiocytes. And like that of mitosis, karyokinesis occurs first, then it is followed by cytokinesis. In karyokinesis of meiosis 1, it is subdivided into prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinesis 1. The prophase 1 of meiosis 1 is one of the longer process and takes more duration in comparison to other phases, as it is subdivided into 5 different substages. First one is leptotin, second zygotin, pachytin, diplotin and diakinesis. In prophase 1, as it has already a longer phase in comparison to other phases, so the first substage is leptotin, and leptotin is characterized by these following points. The chromosome become distinct, as prior to it the chromosome appears indistinct and thread-like, but in this stage the chromosome become distinct. Chromosomes are diploid in number. The chromosome appears like bead-like structures, and centriole starts divide into two centrioles. So it is the first substage of that prophase 1. Then zygotin is the second substage, in which here in this picture, this is the leptotin stage. The chromosome appears like bead-like structure and centriole starts dividing. Here it is not dividing, but it starts dividing, which will be clear in the next stage diagram. So zygotin is characterized by pairing of chromosome occurs by the process of synapsis. The homologous chromosomes that derived from both parents, that is father and mother, they come lie side by side, and by this process it become pairing. That process is called as synapsis, which occurs by presence of a complex called as synaptonemal complex. The pairing of that chromosome is called as bivalent. The paired chromosomes are called as bivalent, and pairing starts from the ends and proceed towards the centromere. From the ends towards the centromere, the pairing continues. the chromosome becomes shorter and thicker. So here the chromosome in leptotin stage become indistinct, but when it become indistinct in comparison to that of zygote stage, as the chromosome becomes shorter and thicker, it become more distinct, and bivalent chromosome is formed due to synapses. The chromosome becomes shorter and thicker, the nucleolus increases its size, and centriole, after its division into two, they move to their opposite poles. Here centromere divides and they travel to their opposite poles. Nucleolus increases its size and the chromosome become bivalent. Then the next substage is phacetin. Each bivalent begin to split. So each bivalent become 4 chromatids, and this stage is called as tetrad. Tetra means 4. So these 2 chromosomes with 4 chromatids, this stage is called as tetrads. Homologous chromosomes of each pair begin to separate due to disappearance of force of attraction between them. So homologous chromosomes do not completely separated but remain attached at certain points. This point is called as chiasma, and X-like structure is cross-like structure is formed, but they remain attached at some points. The chromatids break at points and the broken segments are interchanged. So during interchange of chromosomal segments between 2 non-sister chromatids of homologous chromosome, it is called as crossing over. So during crossing over, interchange of that chromosomal segment takes place, which bring about genetic variation, and that variation bring about genetic recombination. So crossing over between 2 non-sister chromatids always occurs in that homologous chromosome. And the next stage is diplotin. Here in phacetin, the crossing over takes place and the interchange occurs between 2 non-sister chromatids. So genetic recombination takes place there. Diplotin is characterized by the homologous chromosomes repel from each other. Due to less force of attraction between them, they repel from each other. Thus, 2 homologous chromosomes separate each other. By the end of this stage, chiasmata begin to move along the length of chromosome from centromere towards the end. So the division or separation starts from the centromere, which elongates or extends towards the end of the chromosome. The displacement of chiasmata called as terminalization. Here the chiasmata displaced so that the chromatid separates from each other, and that phenomenon is called as terminalization. And last substage of prophase 1 is diakinesis, and it is characterized by the terminalization is completed. So bivalent tend to repel each other and migrate to their periphery of the nucleus, just inside the nuclear membrane. Nucleolus starts to disappear. So here they repel each other and they travel towards the opposite pole. Here one chromosome lies at one pole and another lies at another pole. And nucleolus starts to disappear here in this diagram. Nucleolus is absent, so it starts to disappear. Nuclear membrane is also begin to disappear, and here the spindle fibers are formed from the centriole that remain present on 2 different poles. So spindle fiber make their appearance at the end of prophase 1, that is substage diakinesis. Then it is followed by metaphase 1. The spindle fiber as metaphase is characterized. The chromosomes lies at the equatorial plane or middle plane, and spindle fibers are well developed. Before it, the spindle fibers are formed and it become well developed. Centromere lies towards the pole, but arms of the chromatids lies at the equator. Arms towards the equator, but centromere towards the pole. The two chromatids of each chromosome do not separate. The centromere does not divide. So this is all about that metaphase 1. Then it is followed by anaphase 1. In anaphase 1, each homologous chromosome with its two chromatid and undivided centromere move towards the opposite pole of the cell. Here the chromosome travel towards the opposite pole. So the actual number become reduced. Here two chromosomes are present, here also two chromosomes present, but they travel towards their opposite pole. Here constriction appears, so the chromosome number become haploid. When separated in each chromosome, the sister chromatids are connected by centromere. This stage of chromosome is called as dyad stage. Then it is followed by the last phase of meiosis 1, that is telophase 1. The diploid number of chromosome after reaching their respective poles, chromosome become uncoiled and elongated. The nuclear membrane and nucleus reappear. Thus, two daughter nuclei are formed with haploid number of chromosome. Then it is followed by here this is all about that karyokinesis 1. Then it is followed by cytokinesis 1, in which the cytoplasm makes their constriction at the middle and finally divides that parent cell into two daughter cells with haploid number of chromosome in it. In some cases, the anaphase directly lead to the prophase of homotypic division, so it do not requires that telophase. So then it is undergo that second meiosis or meiosis 2. In meiosis 2, like that of mitosis, it is further subdivided into, one is prophase 2, it is equivalent to that of mitosis. So all the events that occurs in this phase is similar to that of mitosis, so there is no need to describe this, but the events are, first one is prophase 2, then metaphase 2, then anaphase 2 and telophase 2. So this is karyokinesis 2. After completion of that karyokinesis, it is followed by cytokinesis 2. In that cytokinesis 2, each parent cell with diploid number of chromosome in meiosis 1, as it is reduction division, it produces two daughter cells with haploid number of chromosome, but during meiosis 2, it produces 4 daughter cells with haploid number of chromosome. Hence the meiosis 2 is called as equational division. So this is all about that meiosis. If is there any doubt, then you can contact with WhatsApp with me. Thank you.